Hello students, this is Dr. Balding. In this video, we're going to continue talking about our processes of genetics. So, in anatomy and physiology, you probably discussed some of these genetic processes, maybe not in the detail that we're going to cover in this course, but this slide is to remind you about the central dogma of genetics. Right? We call the process where DNA goes to DNA when a cell copies itself as DNA replication. We call the process of going from DNA to RNA transcription, right? And here we're talking about messenger RNA, so that would be the message. And then when RNA is converted into protein, we call that process translation. Okay, this is the central dogma of genetics and was thought to be the only or the way in which genetics worked for many years. That this order was essential, and in most cells, yes, that's true. We do have other processes as well, though, that were discovered later. We have learned since that time that RNA can be converted into DNA in a process that we call reverse transcription that occurs in some viruses. We also know that RNA can be converted into other RNA sequences, and that also occurs in viruses. And when we get to the virus section, we're going to learn more about those processes. So those sort of went against the central dogma of genetics, right? So there are some exceptions to this, line of thinking here, but we're going to talk about those. All right, the first genetic process that we're going to address is that of replication or DNA replication. And this is required when a cell copies itself. It must replicate its DNA so that the daughter cells each have their own copy of the complete genome. So what occurs is that replication occurs in a specific location called the origin. There is one in prokaryotes. There tend to be many in eukaryotes. And it proceeds away from the origin in each direction. Okay? So if you look at this DNA molecule that's shown here, right? Originally, it consisted of, and the picture before that isn't there, a purple single strand of DNA, right? Hydrogen bonded to basically the blue strand. Okay, so that made a double-stranded DNA. And what they're showing here is at a site called the origin. The DNA was opened up by replication enzymes. Those hydrogen bonds were broken. The purple and the blue strand were peeled apart. And what you see is the synthesis of a new beige-looking strand. And so replication is actually proceeding, like I said, in two directions. It's occurring here, right, as the DNA is unzipped, and it's also occurring over here. So there's two replication, and we call them forks, okay? So when this process ends, we have the purple strand served as a template for a daughter strand of DNA that's in beige. And then we have the blue strand served as a template for a daughter strand in beige. And so these would end up in new cells. And so that new cell would contain some old DNA and newly synthesized DNA. We call the usage of the DNA as a template strand and the production of daughter strands that are newly synthesized, this binding of old and new DNA, a semi-conservative form of replication. So one strand is conserved and one strand is new in the daughter cells. All right, so we're going to take a look at this process a little closer. So here is an origin. Like I said, prokaryotes, like bacteria, are going to have one. Eukaryotes, like us, are going to have many. If we didn't have many, we wouldn't be able to replicate our DNA in a timely fashion. So there's usually several along the length of a linear chromosome, but one on the circular double-stranded DNA chromosomes of prokaryotes. Now, so they're showing you where this origin is recognized by replication enzymes and being opened up. So there's just a small region here that's being opened on this circular chromosome, or it could be a plasmid. They use the same mechanism. So then we have a replication fork here, right? So there's one, and there's another replication fork here too. So in the next picture, what I want to show you is just looking at one replication fork, what happens. It's complicated when we're looking at both of them. But I want to go through the entire process with you. All right, so when that origin opens up, hydrogen bonds are being broken between paired nucleobases, right, the nucleotides, and you end up with what we call single-stranded or ssDNA. Right? There are going to be, or there will be, a tendency of those hydrogen bonds to reform. Okay, they're just going to want to come back together like they were before, and so what the cell does is it sends in these enzymes called single-stranded DNA binding proteins, or here they're showing them called stabilizing proteins, that prevent those hydrogen bonds from reforming. It keeps the DNA pretty much single-stranded until this replication process occurs. Now, ahead of the stabilizing proteins are enzymes that unwind the DNA. They're called DNA helicases. Okay, so a helicase functions to unwind. So these two enzymes are unwinding, and then behind them, these DNA stabilizing proteins or single-stranded DNA binding proteins are coming in and preventing those hydrogen bonds from reforming. Okay, so then what happens? Well, there's an enzyme that functions in replicating DNA. It's the primary enzyme called DNA polymerase. Okay, I'm going to circle it here. Now, DNA polymerase is kind of picky, right? It only sees DNA that looks like this, okay? So let me show you. So if we've got a piece of single-stranded DNA, right, it needs to be able to bind to a place where there's a double-stranded region, like that, so there's nucleobases formed there, and it needs to see a 3' hydroxyl. Okay, remember I said we add nucleotides at a 3' hydroxyl end? Well, that end has to be there, right? So DNA polymerase, the only way it's going to see where it needs to go is if it sees something that looks like this. Okay, well, this doesn't exist initially in the DNA. What actually exists is just a single strand, right? It looks like that. So there's another enzyme that we have that comes in first called primase. Okay, primase lays down this little piece here that DNA polymerase needs to get started, right? So its name comes from being a primer. It starts, right? If you've ever, if you want to go paint, right, you might need to prime the walls first for the paint to stick well, right? You prime it. And so this primate lays down this little piece of nucleotide sequence. And it's not actually a DNA sequence, it's actually an RNA sequence that it puts down. Okay? So, DNA polymerase can then see this little piece, and usually it's about 6 to 10 nucleotides long of RNA that's associated with a single-stranded DNA. Okay, it binds, DNA polymerase binds, and then it starts adding new nucleotides to this 3' end. So one there, one there, one there, and so forth. It's doing that dehydration synthesis reaction that we've talked about so many times. And so the chain grows in the 5' to 3' direction. So DNA polymerase only works in that direction. Okay, so that's what you're seeing here, new nucleotides being laid down, and it's going to chase basically the replication fork behind DNA helicase, removing those the stabilizing proteins and adding nucleotides. And that's called leading strand synthesis. Okay? Now, there's something happening on the other strand. Okay, we're not highlighting it here, but DNA polymerase doesn't see or can't work continuously on this other strand. Okay, so at every replication fork, you're going to have 2 strands of DNA. You're going to have one that's being unwound that has a 3' end here and a 5' end, right? And because it's anti-parallel, you're going to have a strand that has a 5' end here and a 3' end here, right? And so as it's being unwound, DNA polymerase is only going to be able to lay down nucleotides in the 5' to 3' direction, going this way, 5' to 3'. Okay, so what does that mean for this synthesis? Well, it means DNA polymerase is going to have to lay nucleotides down on this strand in little pieces. As the DNA is being unwound, okay, it can make a little piece and then it has to wait, or it has to open up and another little piece is unwound, and so forth. And so we call this lagging strand synthesis because we only get little pieces of DNA made at a time. And primase has to work over and over on this strand, laying down these little RNA primers to prime DNA replication. Okay, and it's because DNA polymerase only works in that direction that it can't follow the replication fork, right, that way. No, this can't exist, right? We have to synthesize DNA in that direction. All right, so I'm going to clear the ink a little bit on this diagram so we make it cleaner again. And let's just look at what we have at the end of this process. Well, on this strand, the leading strand, we have a long continuous strand of DNA, but you can imagine at the end of it, when we get all the way around, we're going to have a gap at the end of the process where the DNA is not joined. The sugar phosphate backbone isn't completely made. And then we're also going to have an RNA DNA hybrid there. So there's RNA that the primase laid down paired with a DNA sequence. And then on this strand, we're going to have lots of RNA DNA hybrids, all right, on that lagging strand because it was made discontinuously. Okay, so we have to fix this. All right, the first thing we do is fix the RNA DNA hybrids with another DNA polymerase. It's not the same one that's used for synthesizing the long DNA strands, but there's another one that can come in and fix these RNA DNA hybrids whenever they exist because we don't want to leave them. We don't want just little pieces of RNA throughout our DNA genome. It's not as stable. And then the gaps at the end are fixed by an enzyme called DNA ligase. Okay, it seals the sugar phosphate backbone. So here's an example of DNA ligase sealing a gap like that. right? It's just taking care of those covalent bonds at the end, making sure that the DNA is continuous and there's no gaps left behind. All right, so that's the process of DNA replication. Now, there's some things that I want to mention about DNA polymerase. It's very fast. Okay, DNA polymerase goes at about 1,000 nucleotides per second. Okay, so it's like a jet engine. It's flying through and adding these nucleotides very, very quickly. But it also has the ability to proofread. Okay, so it's got a spell check. So there's this pocket where if nucleotides are mismatched, it can actually back up and chew the DNA that's not right away. So we say it has an exonuclease activity. Its ability to chew that little erroneous piece away, proofread, and then move on. So it gets rid of those mismatches. About 1 in 100,000 nucleotides that DNA polymerase lays down is mismatched and fixed by DNA polymerase. But even, with that, we still get about 1 in a billion remains unfixed. Okay, and we'll talk about repair mechanisms later on that fix the DNA to make sure that we don't end up with a lot of mutations every time DNA is copied, because we don't need mutations. Mutations are typically detrimental. Okay, so another characteristic of the DNA polymerase is it only adds nucleotides to the 3' end of a growing strand. So it works in the 5' to 3' direction. Right? And it also needs a helicase to unwind the DNA because it can't unwind it itself. Okay, and we're going to compare this to RNA synthesis in a little bit, and you'll see how it differs a little bit. All right. So if I were to ask you this question, Okazaki fragments are used to form which DNA strands during replication? Okay, those are the short fragments that were made on the lagging strand, right? And this was the guy that discovered them. Okay, what would you say the answer is? Of course, B here. The lagging strand is responsible, or is the result of DNA polymerase working only in the 5' to 3' direction. Okay, so now there is another aspect of DNA that I want to talk about. DNA can be modified with functional groups. Okay, one type of functional group that can be added to DNA is a methyl group. Remember, methyls were a single carbon with 3 hydrogens, right, attached to some molecule. So DNA can be methylated, and usually it's certain bases that are methylated in DNA, usually specific sequences. Methylation actually is a type of regulation in some sense, or in some cases, and it tells us something about the DNA in that region. In eukaryotes, when genes are highly methylated, right, they are turned off. So when you take off the methyl groups, genes are on. When you add methyl groups, it turns them off. So it's in eukaryotes, it's associated with the ability to switch on or off genes. So in heterochromatin, you usually see a lot of methylation. In euchromatin, you usually see very little methylation. In prokaryotes, on the other hand, methylation does a lot of different things. So how do we know, or how do enzymes know where to identify the start of DNA replication? Okay, the origin is actually highly methylated, right? And that's how those enzymes recognize that particular region of DNA. Also, when we copy DNA in a prokaryote, how do we know, how do repair enzymes know what to fix if there's a mistake? Let's just say we had a sequence that looked like this, right? So we'll just say we've got that sequence and we've got its base pairing, right? It should look something like that. And let's just say that instead of an A here, our DNA polymerase put a C. Okay, so there's a mismatch at this spot. All right, so we've got a big old DNA repair enzyme that comes along and scanning DNA, and it sees this, right? How does it know what to do? Does it put a T where the C was, which would be correct, or will it put a G where the A was? Well, these repair enzymes can detect methylation, and so the old strands of DNA might be methylated, right? So they'll have these methyl groups associated with them along specific sequences. And it'll know that the methylated strand is the old strand and the non-methylated strand is the new strand. Okay, and so repair enzymes will end up fixing this appropriately and putting a T here instead of a G. Right? Also, in prokaryotes, methylation protects against viruses. So let me show you how that happens. So if we have a bacterial cell here, and let's just color its DNA here purple, right? All right, so let's say that a virus, I'm going to draw a little bacteriophage, right, infects the cell and makes copies of more virus. All right. Because the virus takes over the cell and uses this cell's enzymes to synthesize it, right, its DNA is going to look like the DNA of the host, okay, having all the same methylation patterns. Okay, and let's just say this virus goes on to infect another cell, right? And this other cell has a different set of DNA methylases, right? So the viruses that inject their DNA, right, into this host bacterium, well, the bacterium are going to have enzymes that are going to be able to see that DNA and its methylation pattern. And because it's not the same methylation pattern as the host DNA, are going to be able to chew it up and destroy it, right? So they're going to be able to degrade this viral DNA. So it's almost like an immune response of the prokaryotes, their ability to digest DNA from a foreign, or that were made in a foreign host. Okay, so methylation can help protect against viral DNA. All right, so we're going to stop here before the video gets too long, and next we'll talk about transcription.